2. Place a piece of aquatic plant in the indicator in 2 tubes. Try to use similar sizes of plant in each. 3. Use black paper to wrap around one of the tubes with a plant in it, and one without. Make sure the tubes are completely covered. Figure 6.22, using hydrogen carbonate indicator to investigate the effect of light and dark on an aquatic plant. 4. Stand all 4 tubes in the light and leave them for at least 1 hour, preferably longer. 5. When the indicator has changed color in at least one of the tubes without a covering, remove the black paper from the 2 covered tubes. Record the color of the indicator in each tube. Questions. 1. Use the information about the color of hydrogen carbonate indicator to compare the concentration of carbon dioxide in the 4 tubes. 2. Plants, like all living organisms, respire. They do this all the time. Respiration uses oxygen and produces carbon dioxide. In bright light, plants photosynthesize as well as respiring. They photosynthesize faster than they respire. Use this information to explain your results. Limiting factors. If a plant is given plenty of sunlight, carbon dioxide and water, the limit on the rate at which it can photosynthesize is its own ability to absorb these materials and make them react. However, quite often plants do not have unlimited supplies of these materials, and so their rate of photosynthesis is not as high as it might be. A factor that is in short supply, and that therefore limits how quickly the plant can photosynthesize, is called a limiting factor. Light intensity. In the dark, a plant cannot photosynthesize at all. In dim light, it can photosynthesize slowly. As light intensity increases, the rate of photosynthesis increases, until the plant is photosynthesizing as fast as it can. At this point, even if the light becomes brighter, the plant cannot photosynthesize any faster. Figure 6.23, the effect of light intensity on the rate of photosynthesis. Over the first part of the curve in Figure 6.23, between A and B, light is a limiting factor. The low light intensity is preventing the plant from photosynthesizing any faster. The plant is limited in how fast it can photosynthesize because it does not have enough light. You can tell that this is so because the rising line on the graph shows that as the light intensity increases, the rate of photosynthesis increases. Between B and C, however, light is not a limiting factor. You can see this because, even when the light intensity increases, the rate of photosynthesis stays the same. The graph shows that increasing the light intensity beyond B does not increase the rate of photosynthesis. The plant already has as much light as it can use. Carbon dioxide. Carbon dioxide can also be a limiting factor. The more carbon dioxide a plant is given, the faster it can photosynthesize. But once the carbon dioxide concentration reaches a certain level, there is no further increase in the rate of photosynthesis. Figure 6.24, the effect of carbon dioxide concentration on the rate of photosynthesis. Temperature. The chemical reactions of photosynthesis can only take place slowly at low temperatures, so a plant can photosynthesize faster on a warm day than on a cold one. Some of the reactions involved in photosynthesis are catalyzed by enzymes, and therefore temperature affects the rate of photosynthesis in the same way as any enzyme-catalyzed reaction. A graph of rate of photosynthesis against temperature would look quite similar to Figure 5.4. The main difference is that most plant enzymes have an optimum temperature that is much lower than that of mammalian enzymes. The curve would therefore lie to the left of the curve in Figure 5.4. Stomata. Carbon dioxide diffuses into the leaf through the stomata. If the stomata are closed, photosynthesis cannot take place because the plant lacks one of its raw materials. Stomata often close if the weather is very hot and sunny, to prevent too much water being lost. This means that on a really hot or bright day photosynthesis may slow down. Questions. 13. Compare Figure 6.23 with the graph that you drew for your investigation into the effect of light intensity on the rate of photosynthesis. Are they similar in any way? What differences are there? Can you explain these differences? 14. Look at Figure 6.24. Between which values of carbon dioxide concentration is carbon dioxide a limiting factor? Explain how you can tell. Sometimes, you may be asked to think about two limiting factors at once. The graph in Figure 6.25 shows how light intensity affects the rate of photosynthesis at two different carbon dioxide concentrations. Figure 6.25, the effect of light intensity and carbon dioxide concentration on the rate of photosynthesis. First, look at the curve labeled A. As light intensity increases, up to a value of 25 AU, the rate of photosynthesis increases. So, we know that light intensity is a limiting factor over this range. But if light intensity increases above 25 AU, there is no change in the rate of photosynthesis. So light intensity is not a limiting factor now, it is not lack of light that is stopping the plant from photosynthesizing faster. But the graph does show a way of helping the plant to photosynthesize faster at these high light intensities. We can give it more carbon dioxide. Curve B shows the results for a plant that was given a higher concentration of carbon dioxide. Clearly, it is photosynthesizing faster than the one with a low concentration. So, for curve A at high light intensities, carbon dioxide is a limiting factor. Summary. Photosynthesis is a reaction in which plants make carbohydrates from carbon dioxide and water, using light as a source of energy. The word equation for photosynthesis is carbon dioxide plus water glucose plus oxygen. The balanced equation is 6CO plus 6HOCH12O plus 6O. Chlorophyll is present in the chloroplasts of some plant cells. It transfers energy from light into energy in carbohydrates. The carbohydrate glucose is made in photosynthesis, and this can be used to release energy in respiration. Glucose can be used to make starch for storage of energy, cellulose for cell walls, sucrose for transport, and other sugars to make nectar. Glucose can be used to make amino acids and proteins, using nitrogen obtained from nitrate ions. It can also be used to make chlorophyll, using nitrogen, and also magnesium ions. Leaves have a large surface area to capture energy from sunlight and to increase absorption of carbon dioxide from the air. Stomata on the lower surface allow carbon dioxide to diffuse into the air spaces, and then to the cells in the palisade mesophyll layer, where most photosynthesis takes place. To test a leaf for starch, it is first boiled to break down cell membranes, then heated in alcohol to remove the chlorophyll, and finally iodine solution is added. We can use the starch test to show that leaves, or parts of a leaf, that do not have light, chlorophyll or carbon dioxide are not able to photosynthesize and make starch. Controls are needed so that we can compare the results with them. We can use an aquatic plant to measure the rate of photosynthesis, by counting the number of bubbles given off in unit time, or by measuring the volume of gas given off in unit time. This method, using an aquatic plant, can be used to investigate the effect of altering light intensity, temperature or carbon dioxide concentration on the rate of photosynthesis. An environmental factor that is in short supply, and is preventing a plant from photosynthesizing any faster, is called a limiting factor. We can recognize a limiting factor because if the plant is given more of it, the plant photosynthesizes faster. Light intensity, carbon dioxide concentration and temperature can all be limiting factors for photosynthesis. Project: Designing a glass house. Work in a group of 3 or 4 for this activity. You might like to assign different roles to different people. You are going to design a glass house to grow a crop in your country. The glasshouse will provide ideal conditions to help the plants to photosynthesize quickly and grow fast, so that they will provide a high yield for the grower. Your group will then give a presentation to the rest of the class. Figure 6.26, Pepper plants growing in a glasshouse in the Netherlands. These plants are growing with their roots in a liquid containing just the right quantities of the mineral ions that they need, rather than in soil. Here are some things to think about and research. What crop will you grow in the glasshouse? What conditions does this crop need to grow well? How can a glasshouse provide better conditions for the crop than if it was grown outside? How will you control temperature in the glasshouse? In your country, will you need to heat the glasshouse or cool it, or do both at different times? How will you control light in the glasshouse? In your country, will you need to give extra light, or might you need to shade the glasshouse sometimes? How will you provide carbon dioxide in the glasshouse? Providing carbon dioxide costs money, so what might be a suitable concentration to aim for that will give you a good return for your expenditure? You can make your presentation as a video, use PowerPoint, or make models and draw diagrams, or all of these methods if you like. Exam-style questions. 1. What are the raw materials required for photosynthesis? A carbon dioxide and oxygen oxygen, and glucose and water. Water and carbon dioxide. 2. A student investigated the necessity of light for photosynthesis. He covered a leaf with black paper to prevent light reaching it. What is an appropriate control for his experiment? A leaf that has been destarched. B. A leaf that is not covered with black paper. C. A leaf that is partly white and partly green. D. A leaf that has no carbon dioxide provided. 3. Which ions does a plant require to make amino acids from glucose? A calcium, boron, magnesium. Nitrate. 4. In photosynthesis, how many glucose molecules can be made from 30 carbon dioxide molecules and 30 water molecules? A 5, B 6, C 30. D 60. 5. The graph shows how two variables affect the rate of photosynthesis in a plant. At which value of carbon dioxide concentration is carbon dioxide not a limiting factor? 6. The diagram shows a three-dimensional drawing of a section through a leaf. A. I name the tissue labeled C. 2. Outline how this tissue is adapted for its function. B. Give the letters of two cells or tissues that do not contain chloroplasts. C. Describe the function of the cell labeled H. Use the letters on the diagram to describe how water reaches a chloroplast in the leaf. 7. The bar chart shows the mean number of chloroplasts in 4 different types of cell in a leaf. A. Explain why palisade mesophyll cells have so many chloroplasts. B. Which cell could be a cell in the upper epidermis? Explain your answer. C. Guard cells contain chloroplasts, but not as many as spongy mesophyll cells. Which cell could be a guard cell? D. Chloroplasts contain chlorophyll. Describe the function of chlorophyll in photosynthesis. 8. A. Describe how plants synthesize carbohydrates. B. Outline 2 subsequent uses of the carbohydrates made in photosynthesis. 9. An experiment was carried out to measure the rate of photosynthesis of a plant at different temperatures. The experiment was carried out first in normal air, and then in air enriched with carbon dioxide. The results are shown in the graph. A. Use the graph to describe the effect of increasing temperature on the rate of photosynthesis in normal air. B. Explain the differences between the two curves between a temperature of 0 degrees Celsius and 25 degrees Celsius. Use the term limiting factor in your explanation. C. Suggest why the optimum temperature of 25 degrees Celsius is the same for both curves. D. Explain the shape of the curves above 25 degrees Celsius. 10. Two students carried out an experiment to investigate the effect of carbon dioxide concentration on the rate of photosynthesis of an aquatic plant. The photograph shows some of the apparatus that they used. The students filled 5 syringes with 5 different concentrations of sodium hydrogen carbonate solution. This provides carbon dioxide. They placed a piece of aquatic plant in each syringe. They attached a long transparent tube to the nozzle of each syringe. They pushed the plunger of each syringe in a little way to force some of the sodium hydrogen carbonate into the tubing. They then pulled each plunger out a little way to introduce some air into the tubing. They marked the position of the meniscus on each piece of tubing, as shown in the diagram. All 5 syringes were then placed in bright sunshine. As the plants photosynthesized, they released oxygen. The oxygen gas collected above the liquid in the syringe, pushing the liquid down the tube. The students recorded the distance moved by the meniscus in each piece of apparatus after 30 minutes. A. Identify the independent variable and the dependent variable in this investigation. B. Explain why the 5 syringes were placed in bright sunshine. The table shows the results of the experiment. Percentage concentration of sodium hydrogen carbonate solution. Distance moved by meniscus after 30 minutes millimeter. 0, 0.1, 0.2, 0.3, 0.45, 8, 12, 14, 17. See, draw a line graph to display these results. Join the points with ruled straight lines. Dis carbon dioxide a limiting factor at any of the concentrations of sodium hydrogen carbonate used in this investigation. Explain your answer. E. I suggest 2 possible sources of error in this investigation. To suggest how the investigation could be modified to reduce these sources of error. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. I can. See topic. Needs more work. Almost there. Confident to move on. State the word equation for photosynthesis. 6.1. Explain what a limiting factor is. 6.3. Explain how light intensity, temperature, and carbon dioxide concentration can act as limiting factors for photosynthesis. 6.3. Use information presented on graphs to identify limiting factors. 6.3. Chapter 7. Human Nutrition. In this chapter you will learn about a balanced diet, find out about the structure and function of the digestive system, learn how and where physical and chemical digestion happen, practice summarizing information using an annotated drawing. Getting started. First, think on your own about the answers to these questions about biological molecules. Then discuss your ideas with a partner. Be ready to share your ideas. 1. What type of biological molecule is starch? 2. What are the smaller units that a starch molecule is made of? 3. What are the smaller units that a protein molecule is made of? 4. Lipase is an enzyme that digests fats. What are the products of this reaction? 5. What type of biological molecule is lipase? Stomach acid. The photograph in Figure 7.1 shows the inside of a healthy human stomach. The picture was taken using a tiny camera on the end of a tube that can be passed down the esophagus and into the stomach. Figure 7.1, the inside of a healthy human stomach. The stomach is a very important part of the digestive system. It has strong, muscular walls, which help to break up food that you have swallowed and mix it with liquids. The folded walls of the stomach produce a liquid called gastric juice. This juice contains hydrochloric acid and enzymes that break down protein molecules. The hydrochloric acid in your stomach has a concentration of about 0.1 moles dm. Hydrochloric acid is a strong acid. If you dipped a piece of blue litmus paper into this acid, it would go red. The acid helps us by killing many of the microorganisms in the food that we swallow. The stomach does not produce acid all the time. It starts to do it when we smell, taste, see or think about eating food. Discussion questions. 1. The stomachs of some carnivorous animals, such as lions and hyenas, secrete more concentrated acid than we do. Can you suggest how this may help them to survive? 2. Suggest why it is not good for the stomach to secrete acid all the time. 7.1 Diet. The food an animal eats every day is called its diet. Humans need 6 types of nutrient in their diet. These are carbohydrates, proteins, fats, vitamins, minerals, water. In addition, the diet should contain fiber. This is not really a nutrient, as it is not absorbed into the body. It just passes straight through the digestive system and is passed out in the feces. A diet which contains all of these things, in the correct amounts and proportions, is called a balanced diet. Energy needs. Every day, a person uses up energy. The amount you use partly depends on how old you are, which sex you are, and what job you do. A few examples are shown in Figure 7.2. Figure 7.2, Daily Energy Requirements. The energy you use each day comes from the food you eat. If you eat too much food, some of the extra will probably be stored as fat. If you eat too little, you may not be able to obtain as much energy as you need. This will make you feel tired. All food contains some energy. Scientists have worked out how much energy there is in particular kinds of food. You can look up this information on the internet. 1 gram of fat contains about twice as much energy as 1 gram of protein or carbohydrate. A person's diet may need to change at different times of their life. For example, a woman will need to eat a little more each day when she is pregnant, and make sure that she has extra calcium and iron in her diet to help to build her baby's bones, teeth and blood. She will also need to eat more while she is breastfeeding. Most people find that they need to eat less as they reach their 50s and 60s because their metabolism slows down. Nutrients. As well as providing you with energy, food is needed for many other reasons. To make sure that you eat a balanced diet, you must eat foods containing carbohydrate, fat and protein. You also need each kind of vitamin and mineral, fiber and water. If your diet doesn't contain all of these substances, your body will not be able to work properly. Carbohydrates. Carbohydrates are needed for energy. Carbohydrates include starch and sugar. In most countries, there's a staple food that supplies much of the carbohydrate in people's diets, in the form of starch. Staple foods include potatoes, wheat, rice and maize. We also eat carbohydrate in sweet foods, which contain sugar. Figure 7.3, Some good sources of carbohydrate. Fats and oils. Fats and oils are needed for energy, and to make cell membranes. We store excess fat and oil under the skin, in adipose tissue. Here, it acts as an insulator, reducing heat loss from the body to the air. It can also form a layer around body organs such as the kidneys, providing mechanical protection for them. We obtain fat from cooking oils, meat, eggs, dairy products and oily fish. Figure 7.4, Some good sources of fats and oils. Proteins. Proteins are needed to build new cells, for growth. They are also used to make proteins, including hemoglobin, insulin, and antibodies. We get protein from meat, fish, eggs, dairy products, peas and beans, nuts and seeds. Figure 7.5. Some good sources of protein. Vitamins. Vitamins are organic substances, which are only needed in tiny amounts. If you do not have enough of a vitamin, you may get a deficiency disease. Table 7.1 shows information about sources and uses in the body of vitamins C and D. Vitamin foods that contain it. Why it is needed. Deficiency disease. C. Citrus fruits, raw vegetables. To make the stretchy protein collagen, found in skin and other tissues, keeps tissues in good repair. Scurvy, which causes pain in joints and muscles, and bleeding from gums and other places. This used to be a common disease of sailors, who had no fresh vegetables during long voyages. D. Butter, egg yolk. Helps calcium to be absorbed, for making bones and teeth. Rickets, in which the bones become soft and deformed. This disease was common in young children in industrial areas, who rarely got out into the sunshine. Table 7.1, Vitamins C and D. Minerals. Minerals are inorganic substances. Once again, only small amounts of them are needed in the diet. Table 7.2 gives information about the sources and uses of two of the most important ones. Mineral ion foods that contain it. Why it is needed. Deficiency disease. Calcium. Milk and other dairy products, bread. For bones and teeth, for blood clotting. Brittle bones and teeth, poor blood clotting. Iron. Liver, red meat, egg yolk, dark green vegetables. For making hemoglobin, the red pigment in blood which carries oxygen. Anemia, in which there are not enough red blood cells so the tissues do not get enough oxygen delivered to them. Table 7.2, Calcium and iron. Fiber. Fiber helps to keep the alimentary canal working properly. The alimentary canal is the part of the digestive system through which food passes, as it moves from the mouth to the anus. Food moves through the alimentary canal because the muscles contract and relax to squeeze it along. This is called peristalsis. The muscles are stimulated to do this when there is food in the alimentary canal. Soft foods do not stimulate the muscles very much. The muscles work more strongly when there is harder, less digestible food, like fiber, in the alimentary canal. Fiber keeps the digestive system in good working order and helps to prevent constipation. All plant foods, such as fruits and vegetables, contain fiber. This is because plant cells have cellulose cell walls. Humans cannot digest cellulose. Figure 7.6. Some good sources of fiber. One excellent source of fiber is the outer husk of cereal grains, such as oats, wheat and barley. This is called bran. Some of this husk is found in wholemeal bread. Brown or unpolished rice is also a good source of fiber. Water. More than 60% of the human body is water. Water is an important solvent. Cytoplasm is a solution of many substances in water. The spaces between our cells are also filled with a watery liquid. Inside our cells, chemical reactions are happening all the time. These are called metabolic reactions. They can only take place if the chemicals that are reacting are in solution. If a cell dries out, then the reactions stop, and the cell dies. The liquid part of blood, plasma, is also mostly water. It contains many substances dissolved in it, which are transported around the body in the blood. Water is also needed to dissolve enzymes and nutrients in the alimentary canal, so that digestion can take place. We also need water to get rid of waste products. As you will see in Chapter 13, the kidneys remove a waste product called urea from the blood. The urea is dissolved in water to form urine. We get most of our water by drinking fluids, but some foods such as fruit also contain a lot of water. Activity 7.1. A balanced diet in your country. You are going to work as a group to construct a balanced diet by thinking about foods that are commonly eaten in your country. In your group, discuss the foods that people eat in your country. Decide which ones are good sources of each kind of nutrient. Construct a simple menu for the meals someone will eat in a day, ensuring that all the different nutrients are contained in the food. Don't forget to include drinks as well. If you can, collect these foods together and take photographs of the meals, or the ingredients from which they would be made. Questions. 1. Too much animal fat in the diet can increase the risk of heart disease. Explain why, despite this, we still need to eat some fat or oil. 2. Too much sugar in the diet can increase the risk of tooth decay and diabetes. Explain how we can avoid these risks, but still eat plenty of carbohydrate. 3. Construct a table showing the main dietary sources of carbohydrate, fat and protein, and their uses in the body. Reflection. Some people find that summarizing facts in a table, as in question 3, helps them to learn and remember the information. Does that work for you? Is it better if you construct the table yourself, or if you just learn a table that is given to you? 7.2. The human digestive system. You may remember that a body system, such as the digestive system, is a group of organs that work together to perform a particular function. The human digestive system includes the alimentary canal, and also the liver and the pancreas. All of these work together to break down the food that we eat, so that it can be absorbed into the blood and delivered to all body cells. The digestive system deals with the food in a series of processes. These are: Figure 7.7. How the digestive system of a fox deals with food. The human digestive system works in a similar way. Ingestion, this means taking food and drink into the mouth. We do this using the lips, teeth and tongue. Digestion, the food that we eat contains large pieces and large molecules. The large pieces have to be broken down to small pieces, and the large molecules must be broken down to small molecules. The small molecules can then get through the walls of the intestines and into the blood. Absorption, this is the movement of the small nutrient molecules and mineral ions through the walls of the intestines and into the blood. Assimilation, this is what happens to the nutrients as they become part of the body. They are absorbed by individual cells and used for energy or to make new substances. Digestion, there is always some material in our food that we cannot digest. Much of this is fiber. This is not absorbed. It remains in the intestines and eventually passes out as feces. The processes of chemical digestion and physical digestion are described in Topic 7.3. Absorption is described in Topic 7.4. To begin with, we will look at the structure of the alimentary canal and outline what happens in each part. The alimentary canal. The alimentary canal is a long tube which runs from the mouth to the anus. It is part of the digestive system. As we have seen, the digestive system also includes the liver and the pancreas. The wall of the alimentary canal contains muscles, which contract and relax to make food move along. These muscular contractions are called peristalsis. Sometimes, it is necessary to keep the food in one part of the alimentary canal for a while before it is allowed to move to the next part. Special muscles can close the tube completely in certain places. They are called sphincter muscles. To help the food to slide easily through the alimentary canal, it is lubricated with mucus. Mucus is made in goblet cells, which are found in the lining of the alimentary canal, along its entire length. Each section of the alimentary canal has its own part to play in the digestion, absorption and digestion of food. Figure 7.8 shows the main organs of the digestive system. Figure 7.8. the human digestive system. Questions. These questions are about Figure 7.8. 4. Explain the difference between the alimentary canal and the digestive system. 5. Name 2 parts of the digestive system that food does not pass through. The mouth. Food is ingested using the teeth, lips and tongue. The teeth then bite or grind the food into smaller pieces, increasing its surface area. This begins physical digestion. You can find much more about teeth in Topic 7.3. The tongue mixes the food with saliva and forms it into a little ball that can be swallowed. The salivary glands make saliva. This is a mixture of water, mucus and the enzyme amylase. The water helps to dissolve substances in the food, allowing us to taste them. The mucus helps the chewed food to bind together to form a small ball, and lubricates it so that it slides easily down the esophagus when it is swallowed. Amylase begins to digest starch. See Topic 7.3 for much more about this, and all the other examples of digestion mentioned here. The esophagus. There are 2 tubes leading down from the back of the mouth. The one in front is the trachea or windpipe, which takes air down to the lungs. Behind the trachea is the esophagus, which takes food down to the stomach. The hole in the center of the esophagus, down which food can pass, is called a lumen. This word is used to describe the space in the middle of any tube in the body. There is a lumen in all parts of the alimentary canal. All blood vessels also have a lumen. The entrance to the stomach from the esophagus is closed by a sphincter muscle. The muscle relaxes to let food pass into the stomach, then contracts to close the entrance again. The stomach. The stomach has strong, muscular walls. The muscles contract and relax to mix the food with the enzymes and mucus. Figure 7.9. This artwork shows where some of the organs of the digestive system are positioned in the body. Can you name any of them? Like all parts of the alimentary canal, the stomach wall contains goblet cells which secrete mucus. It also contains other cells which produce enzymes and others which make hydrochloric acid. The enzymes produced in the stomach digest proteins, so they are proteases. The hydrochloric acid produces a low pH, of about pH 2, in the stomach, which kills harmful microorganisms in the food. This pH is also the optimum pH for the protease enzymes that act in the stomach. The stomach can store food for quite a long time. After 1 or 2 hours, the sphincter at the bottom of the stomach opens and lets the partly digested food move into the duodenum. The small intestine. The small intestine is the part of the alimentary canal between the stomach and the colon. It is about 5 m long. It is called the small intestine because it is quite narrow. Different parts of the small intestine have different names. The first part, nearest to the stomach, is the duodenum. The last part, nearest to the colon, is the ileum. The pancreas is a cream-colored gland, lying just underneath the stomach. A tube called the pancreatic duct leads from the pancreas into the duodenum. Pancreatic juice is a fluid made by the pancreas. It flows along the pancreatic duct into the duodenum. Pancreatic juice contains many different enzymes, so chemical digestion continues in the duodenum. The ileum is where all the digested nutrients are absorbed into the blood. This is described in Topic 7.4. Water is also absorbed into the blood at this stage. The large intestine. The final part of the alimentary canal is the large intestine. It is made up of the colon and rectum. The colon absorbs much of the water that still remains in the food. The rectum stores undigested food as feces. These are then egested from the body through the anus. The pancreas and liver. The pancreas and liver are important organs in the digestive system, but they are not part of the alimentary canal. Food does not pass through them. We have seen that the pancreas secretes a fluid containing enzymes, which help with digestion of food in the duodenum. The liver also secretes a fluid that helps with digestion. This fluid is called bile. The bile that has been made by the liver is stored in the gallbladder. When food enters the duodenum, the bile flows along the bile duct and is mixed with the food in the duodenum. Bile is a yellowish-green, alkaline, watery liquid, which helps to neutralize the acidic mixture from the stomach. Questions. 6. List, in order, all the parts of the digestive system through which food passes, as it moves from the mouth to the anus. 7a. Which of the parts you have named in question 6 are involved in ingestion? B. Which parts are involved in digestion? C. Which parts are involved in absorption? D. Which parts are involved in digestion? 7.3. Digestion. The food that we eat usually contains large molecules of starch, protein and fat. These large molecules are too big to get through the walls of the intestines and into the blood. They must be broken down into much smaller molecules, allowing them to be absorbed. As you have seen, this process is called digestion. Digestion takes place in 2 stages. Physical digestion. First, large pieces of food are broken down into smaller pieces. This is mostly done by the teeth, but the churning movements of the stomach also help. This physical digestion does not change the chemical components of the food in any way. Chemical digestion. Next, the large molecules in the food are broken apart into smaller molecules. This involves chemical reactions, and they are catalyzed by enzymes. These smaller molecules are soluble in water, which makes it easier for them to be absorbed into cells. Table 7.3 summarizes what happens to each of the large molecules of starch, protein and fat as a result of digestion. The molecules of vitamins and water are already small enough to be absorbed, and this is also the case for mineral ions. So, none of those need to be chemically digested. Large molecule. Enzyme that breaks it down. Small molecules produced. Starch. Amylase. Simple reducing sugars protein. Protease. Amino acids fat. Lipase. Fatty acids and glycerol. Table 7.3, the effect of digestion on starch, protein and fat. Teeth. Teeth help with the ingestion and mechanical digestion of the food we eat. Teeth can be used to bite off pieces of food. They then chop, crush or grind the pieces into smaller pieces. This gives the food a larger surface area, which makes it easier for enzymes to work on the food in the digestive system. It also helps any soluble molecules or ions in the food to dissolve in the watery saliva in the mouth. The structure of a tooth is shown in Figure 7.10. The tooth is embedded in the gum. The part of the tooth above the gum is covered with enamel. Enamel is the hardest substance made by animals. It is very difficult to break or chip it. However, it can be dissolved by acids. Bacteria feed on sweet foods left on the teeth. These bacteria produce acids, which dissolve the enamel and cause decay. Figure 7.10, the structure of an incisor tooth. Under the enamel is a layer of dentine, which is rather like bone. Dentine is quite hard, but not as hard as enamel. It has channels in it which contain living cytoplasm. In the middle of the tooth, there are nerves and blood vessels. The blood vessels supply the cytoplasm in the dentine with nutrients and oxygen. The part of the tooth that is embedded in the gum is covered with cement. This has fibers growing out of it. These attach the tooth to the bone in the jaw, but allow it to move slightly when biting or chewing. Most mammals have 4 kinds of teeth. Incisors are the sharp-edged, chisel-shaped teeth at the front of the mouth. They are used for biting off pieces of food. Canines are the more pointed teeth at either side of the incisors. Premolars and molars are the large teeth towards the back of the mouth. They are used for chewing food. In humans, the ones right at the back are sometimes called wisdom teeth. They do not grow until much later in the person's development than the others. Figure 7.11, the 4 types of mammalian teeth. Activity 7.2. Counting your own teeth. You can work on your own for this activity, using a mirror to look at your own teeth. Alternatively, you could work with a partner and count each other's teeth. Count the total number of teeth on your upper jaw. Then count how many of these are incisors, canines or premolars and molars. Now do the same for your lower jaw. Does everyone in your class have the same number of teeth as you? If not, suggest reasons for any differences. Chemical digestion. The actions of the teeth and stomach help to break food into smaller pieces, increasing its surface area. This means enzymes can more easily make contact with the molecules in the food and start to break them apart. Table 7.4 summarizes where each type of enzyme is made, and where it works in the human digestive system. Enzyme where it is secreted. Where it acts. Amylase. By the salivary glands. In the mouth. By the pancreas. In the duodenum. Protease. By the walls of the stomach. In the stomach by the pancreas. In the duodenum. Lipase. By the pancreas. In the duodenum. Table 7.4. The sites of secretion and action of digestive enzymes. In Chapter 5, we saw that enzymes are affected by temperature and pH. The digestive enzymes in the human digestive system all work best at a temperature of about 38 degrees Celsius, which is our normal body temperature. The digestive enzymes vary in the pH at which they work best. The stomach walls secrete hydrochloric acid to kill bacteria, so the protease that works in the stomach has to be able to work in these conditions. It has an optimum pH of about 2. The enzymes that work in the duodenum require a pH of just above 7 to work at their fastest. This higher pH is provided by bile and pancreatic juice, which contain alkaline substances that neutralize the acidic mixture that passes into the duodenum from the stomach. Questions. 8 teeth are involved in physical digestion. A. How do they do this? B. Explain how physical digestion helps chemical digestion to happen faster. 9. Describe how the structure of a molar tooth adapts it for its function. 10. State 2 places in the human digestive system where starch is broken down to simple reducing sugars. 11. State 2 places in the human digestive system where proteins are digested. Enzymes in the human digestive system. In this topic, we will look in more detail at the way that digestive enzymes break down large food molecules. We have seen that amylase is secreted into the mouth and the duodenum. The amylase in these 2 parts of the digestive system does the same thing. It breaks starch molecules down to a sugar called maltose. You may be able to taste this sugar if you chew starchy food for a long time, as the amylase in your saliva breaks the starch down. Each maltose molecule is made of 2 glucose molecules linked together. Although maltose molecules are much smaller than starch molecules, they are still too big to be absorbed. Another enzyme, maltase, breaks the maltose molecules apart to produce glucose molecules. Maltase is secreted by the cells lining the small intestine. This lining tissue is called an epithelium. The maltase does not go into the lumen of the small intestine, but remains attached to the epithelial cells, sitting on their cell membranes. So, maltose is digested on the surface of these epithelial cells. We can summarize this 2-step process like this: amylase, starch, maltose, glucose. Protease is secreted into the stomach and the duodenum. The protease that is produced in the stomach is called pepsin. Pepsin is secreted from the stomach walls, in a liquid called gastric juice. Gastric juice also contains hydrochloric acid, to kill harmful microorganisms in food. Pepsin has an optimum pH of 2, so it is perfectly suited to working in these very acidic conditions. The protease that is produced in the pancreas, and works in the duodenum, is called trypsin. Pepsin and trypsin both break protein molecules apart, producing amino acids. Amino acids are small enough to be absorbed. Trypsin has an optimum pH just above 7. As we have seen, this means that the acidic contents of the stomach must be neutralized, and this is done by the alkaline substances in bile and pancreatic juice. Bile has another important function. Fats and oils are insoluble in water, so any fat or oil in the food that you eat stays as little drops that do not fully mix into the watery fluids inside the digestive system. Bile acts rather like liquid detergent. It breaks up large drops of fat or oil into tiny droplets, which can disperse among the watery liquids. A mixture of tiny, floating droplets of one liquid in another is called an emulsion. So, bile emulsifies fats. Figure 7.12, a summary of physical and chemical digestion in the human digestive system. Notice that bile does not actually do anything to the individual fat molecules. Emulsification is physical digestion, not chemical digestion. Emulsification increases the surface area of the fat droplets. So, the emulsified fats are much easier for the fat-digesting enzyme, lipase, to access. Lipase can now chemically digest the fats, breaking them apart into fatty acids and glycerol. Figure 7.12 summarizes the physical and chemical digestion of fats, proteins and carbohydrates in the human digestive system. Questions. 12. Name the part of the digestive system where each of these events occurs. A. Amylase breaks down starch to maltose. B. Maltase breaks down maltose to glucose. 13. A. From which part of the digestive system is lipase secreted? B. In which part of the digestive system does lipase act? 14. A. Where is pepsin secreted? B. Explain why pepsin stops working when the contents of the stomach pass into the duodenum. 15. Bile emulsifies fats. Explain what this means, and why it is an important part of fat digestion. Activity 7.3. Functions of organs in the digestive system. You can work in a group of 2 or 3 for this activity. On a large sheet of paper, make a very big drawing of the human digestive system. Leave plenty of space around your drawing for labels and annotations. Label each part. To each label, add a description of what happens in that part. You could use different pens to color code your written descriptions. For example, you could use one color to represent physical digestion and a different color to represent chemical digestion. Note: after you have learnt about absorption, in the next topic in this chapter, you may like to return to your drawing and add some more information about the small intestine. So, try to leave a little bit of space for this. Peer assessment. Exchange your finished drawing with another group. Compare your drawing with theirs. Which one is clearer? Which contains more information? Which is the more interesting? 7.4 Absorption and Assimilation. By the time food has passed through the duodenum, most of the large molecules have been fully digested to small ones. Carbohydrates have been broken down to glucose, proteins to amino acids, and fats to fatty acids and glycerol. These small, soluble molecules are now able to pass through the walls of the small intestine and into the blood. This process is called absorption. There are also several other small molecules and ions in the food, which were already small enough to be absorbed and did not need to be digested. Mineral ions, vitamins and water are all absorbed into the blood from the small intestine. A smaller quantity of water is also absorbed in the colon. Villi. The inner wall of the small intestine is covered with tiny projections. They are called villi. Each villus is about 1 mm long. Figure 7.13 shows the lining of the small intestine with thousands of villi. Figure 7.13, the villi that line the small intestine greatly increase its internal surface area. Figure 7.14 shows the structure of a single villus. Figure 7.14, the structure of a villus. The cell membrane on the surface of each villus is folded to form many tiny microvilli. It is on these membranes that maltase acts, breaking down maltose into glucose molecules. It is also through these membranes that glucose, amino acids, fatty acids, glycerol, vitamins, mineral ions and water are absorbed. Most of these substances pass into the blood capillaries inside the villus. The blood capillaries from all the villi link up to join a vein, called the hepatic portal vein, which takes all of these substances to the liver. The liver cells absorb and assimilate these substances. Glucose may be changed by the liver cells to glycogen for storage. You can read more about this in Chapter 13. Amino acids are used by the liver cells to make many different kinds of proteins, some of which are then returned to the blood to be transported to other parts of the body. If there are more amino acids than the body requires, they may be broken down in the liver to form urea, which is excreted. This is also described in Chapter 13. The fatty acids and glycerol pass into the lacteals. The contents of the lacteals are eventually emptied into the blood. You can see from the image in Figure 7.13 that the villi hugely increase the surface area of the lining of the small intestine. And as each villus is covered with thousands of microvilli, the total surface area is increased even more. This greatly speeds up the rate at which absorption happens. Summary. A balanced diet contains some of all 6 nutrients: proteins, carbohydrates, fats, minerals, vitamins and water in suitable amounts and proportions. It also contains fiber. Scurvy is caused by a lack of vitamin C, and rickets by a lack of vitamin D. The digestive system is made up of the mouth, esophagus, stomach, duodenum and ileum. Colon and rectum, the liver, gallbladder and pancreas. Food is first ingested. This is followed by digestion, absorption and assimilation. Food that cannot be absorbed is egested. Physical digestion is done by the teeth and stomach. It involves breaking large pieces of food to small ones. Chemical digestion is done by enzymes in the mouth, stomach and small intestine. It involves breaking down large molecules of nutrients to small ones, so that they can be absorbed. Teeth help in physical digestion, grinding food into smaller pieces to increase its surface area. This allows subsequent chemical digestion to happen faster. Humans have 4 types of teeth: incisors, canines, premolars and molars. In chemical digestion, amylase breaks down starch to simpler reducing sugars, protease breaks down proteins to amino acids, lipase breaks down fats to fatty acids and glycerol. Hydrochloric acid in the stomach provides a low pH for enzymes to work, and also kills harmful microorganisms in food. Amylase breaks down starch to maltose in the mouth and duodenum. Maltase breaks down maltose to glucose, on the surface of the villi in the small intestine. The protease in the stomach is called pepsin, and it requires a low pH, which is supplied by the hydrochloric acid secreted in gastric juice. The pancreas secretes a protease called trypsin. This requires a higher pH, which is provided by alkaline substances in bile. Bile is secreted by the liver and stored in the gallbladder before being released into the duodenum. It emulsifies fats, which makes it easier for lipase to digest them. Nutrients, including water, are absorbed into the blood from the small intestine. More water is absorbed from the colon. Villi and microvilli increase the surface area of the small intestine, which speeds up absorption. Project: Make a model of the human alimentary canal. You are going to work in a group to make a model of the human digestive system. You can use whatever materials you can find. For example, you may like to use cardboard tubes of various sizes, balloons, whatever you think will work well for you. Be imaginative. First, discuss in your group how you will construct your model. Think about the materials that you will need. Gather your materials together and make your model. You will almost certainly make some changes to your original plan as you work. That is fine. When everyone has completed their models, they can be displayed around the room. 1 or 2 people from each group should stay with their model and explain it to others as they walk around the display. Then swap over, so that everyone has a chance to see all the other models, and to help to explain their own model. Figure 7.15. Students planning how to model the human digestive system. Exam-style questions. 1. What causes scurvy? A. Lack of calcium in the diet. B. Lack of vitamin C in the diet. C. Much carbohydrate in the diet. D. Much fat in the diet. 2. Where in the digestive system are proteins enzymes secreted? A. Mouth and stomach. B. Stomach and pancreas. Pancreas and liver. D. Liver and mouth. 3. From which part of the digestive system is most water absorbed into the blood? A. Large intestine. Mouth. Small intestine. D. Stomach. 4. What is assimilation? A. Breakdown of large molecules into small molecules. B. The movement of small molecules from the intestine into the blood. C. The movement of small molecules into cells, where they are used. D. The taking of food into the mouth. 5. What is a function of bile? A. Chemically digesting fats, emulsifying proteins, killing harmful microorganisms. D. Increasing pH. 6. The diagram shows the human digestive system. A. Name the parts labeled G and J. B. Give the letters of 2 parts where amylase is secreted. C. Give the letter of 1 part where lipase acts. D. Describe how part C helps in digestion. 7. The diagram shows the teeth in the upper jaw of a human. A. Name the types of tooth labeled A, B and C. B. Outline the functions of tooth A and tooth C. C. Draw and label a diagram to show the internal structure of the tooth labeled C. B. Outline the importance of carbohydrate in the diet. C. Describe how and where carbohydrates are chemically digested in the human digestive system. 9. The photograph shows a section through 2 villi from the human digestive system. A. Name the part of the human digestive system in which villi are found. B. Name the part of the villus labeled A and describe its functions. C. The cell labeled B secretes mucus. Suggest the function of mucus in this part of the digestive system. D. The part labeled C is a blood capillary. Name 2 nutrients that are absorbed into the blood capillaries in a villus. E. Some nutrients are not absorbed into blood capillaries. Name the structures into which these nutrients are absorbed. 8. State 2 principal sources of carbohydrate in a balanced diet. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. I can see topic. Needs more work. Almost there. Confident to move on. Explain what is meant by a balanced diet. 7.1. Give examples of good sources of each type of nutrient, and the importance of each nutrient. 7.1. Identify the main organs of the digestive system. 7.2. Describe the functions of each of the organs in the digestive system. 7.2. Explain the difference between physical digestion and chemical digestion. 7.3. Explain why digestion has to happen before nutrients can be absorbed. 7.3. Describe the different types of teeth, the structure of a tooth, and the functions of teeth. 7.3. State where amylase, protease and lipase act, and what they do. 7.3. Describe the function of hydrochloric acid in the stomach. 7.3. State that the small intestine is where most nutrients are absorbed, including water, and that more water is absorbed in the colon. 7.4. Describe where amylase, maltase, pepsin and trypsin act, and what they do. 7.3. Explain the functions of bile, including providing an alkaline pH and emulsifying fats. 7.3. Describe the structure of a villus, and explain how villi and microvilli speed up absorption. 7.4. Chapter 8. Transport in plants. In this chapter you will learn about the functions of xylem and phloem, and where they are found. Describe the pathway taken by water as it moves through a plant. Investigate the movement of water through stem and leaves. Find out what transpiration is, why it happens, and conditions that affect its rate. Learn about translocation of sucrose and amino acids in plants. Getting started. Work in a group of 3. In your group, think of a way to complete each of these sentences. Try to include some good scientific information in your sentence. Plants take up water. Water gets to the leaves of plants. When each group has completed the sentences, share the different ideas from all the groups. Work together to combine everyone's ideas into 2 sentences summarizing what you already know about how leaves obtain water. The world's tallest trees. Is there any limit to the height to which a tree can grow? The world's tallest trees are the coastal redwoods, Sequoia sempervirens, that can be found in some parts of California in the USA. The tallest one is 116 m tall, growing in the Redwood National Park. This tree is probably 2,500 years old. Figure 8.1, resistance to fire, has allowed redwood trees to live for thousands of years. Scientists think that it is impossible for a tree to grow taller than about 130 m. This is because of the xylem that makes up most of a tree's trunk. Xylem is the tissue that wood is made of. It contains dead, empty cells joined end to end, called xylem vessels. These long tubes run all the way up through a tree's trunk and out into its branches. Xylem vessels have walls made of a very strong substance called lignin. These vessels have 2 functions: they help to hold the tree up, and they provide a pathway for water to flow from the roots all the way up to the leaves at the top of the tree. Discussion questions. 1. Small plants, such as mosses, do not have xylem vessels to transport water. How might water get to all the different cells of a moss plant? 2. It takes about 10 minutes for water to move by osmosis through a cylinder of potato that is 1 centimeter in diameter. What does this suggest about the possibility of using osmosis alone to supply the leaves at the top of a giant redwood tree? 8.1 Xylem and phloem. Plant transport systems. All organisms need to obtain substances from their environment. For plants, these substances are carbon dioxide and water for photosynthesis, and mineral ions which they absorb from the ground. Plants have branching shapes. This gives them a large surface area in relation to their volume. It means that most cells are close to the surface. As we saw in Chapter 6, leaves are adapted to ensure that no cell is far away from the air, so carbon dioxide can simply diffuse in through the stomata and air spaces, easily reaching the photosynthesizing mesophyll cells. Water comes from further away. Plants absorb water through their roots, and this water must be transported up to the leaves. The transport system that does this is made up of a tissue called xylem. Mineral ions are also transported in xylem. Plants also have a second transport system, made up of a tissue called phloem. Phloem transports sucrose and amino acids from the leaves where they are made, to other parts of the plant such as its roots and flowers. Xylem. A xylem vessel is like a long drain pipe. It is made of many hollow, dead cells, joined end to end. The end walls of the cells have disappeared, so a long, open tube is formed. Xylem vessels run from the roots of the plant, right up through the stem. They branch out into every leaf. Figure 8.2, diagrams showing the structure of xylem vessels. Figure 8.3, this scanning electron micrograph shows xylem vessels in a plant stem. The parts colored orange are made of lignin. The biggest xylem vessel is about 100 micrometers in diameter. Xylem vessels contain no cytoplasm or nuclei. Their walls are made of cellulose and lignin. Lignin is very strong, so xylem vessels help to keep plants upright. Wood is made almost entirely of lignified xylem vessels. Table 8.1 summarizes how the structure of xylem vessels is related to their functions. Function feature. Explanation. Support. The walls of vessels contain lignin. Lignin is very strong and can support the great weight of even a heavy tree. In leaves, xylem vessels in vascular bundles help the leaf to be held flat, to provide a large surface area to absorb sunlight. Transport. The cells are dead, with no contents. Water can flow easily through the tube. There are no cross walls between the dead cells. There is a continuous tube for water to flow through, all the way from the roots to the leaves. The walls of vessels contain lignin. Lignin is solid and makes sure the vessels stay open and do not collapse, allowing water to flow through easily. The diameter of the vessels is between about 15 micrometers and 200 m. This is narrow enough to make sure the column of water inside them does not break, but wide enough to allow a lot of water to flow through. Table 8.1, Structure and Function of Xylem Vessels. Activity 8.1. Identifying the positions of xylem vessels in stems, roots and leaves. You are going to use micrographs to make simple diagrams. Your diagrams will show the position of xylem vessels in a stem, a root and a leaf. Figure 8.4. Scanning electron micrograph of a section across a plant stem. One look at Figure 8.4. It shows a plant stem, cut across. The xylem vessels have been colored blue. 2. Draw a circle to represent the section across the stem. Then draw triangles to show where the xylem vessels are found. Label the xylem on your drawing. 3. Look at Figure 8.5. It shows a plant root, cut across. The xylem vessels are the big, empty cells with red walls in the center. The walls are red because a red stain has been used to make the lignin show up clearly. Figure 8.5, Photomicrograph of a section across a plant root. 4. Draw a diagram, similar to the one that you drew of the stem, to show the position of xylem vessels in a root. 5. Look at Figure 8.6. It shows part of a plant leaf, cut across. A stain has been used to color the cell walls. The xylem vessels are the empty cells with purple walls, arranged in a semicircle. Figure 8.6, Photomicrograph of a section across a plant leaf. 6. Draw a diagram, similar to the ones that you drew of the stem and root, to show the position of xylem vessels in a leaf. Self-assessment. How well did you achieve each of these goals? Rate yourself according to the following scheme for each of the features of your drawing listed opposite. If you did it really well. If you made a good attempt at it and partly succeeded if you did not try to do it, or did not succeed. Your drawings are large, bigger than the size of the micrographs. You used a sharp pencil for your drawings. The lines in your drawings are single and unbroken. You showed the position of the xylem correctly and clearly. You labeled the xylem with a label line drawn with a ruler, touching the xylem. Vascular bundles. Xylem vessels and phloem tubes are usually found close together. A group of xylem vessels and phloem tubes is called a vascular bundle. The positions of vascular bundles in roots and stems are shown in figures 8.7 and 8.8. In a root, vascular tissue is found at the center. In a stem, vascular bundles are arranged in a ring near the outside edge. Vascular bundles are also found in leaves. They help to support the leaves, holding them out flat to capture sunlight. Figure 8.7, diagram of a transverse section of a root. Figure 8.8, diagram of a stem cut across, to show the positions of xylem and phloem tissue. Questions. 1. State the two functions of xylem. 2. Name the substance that makes up the cell walls of xylem vessels. 3. State the function of phloem. 4. Use your diagrams from Activity 8.1, and the diagrams in Figures 8.7, 8.8 and 6.11, to describe in words the positions of xylem and phloem in roots, stems and leaves. 5. Explain 2 ways in which the structure of xylem vessels is related to their functions. 8.2 Transport of water. Water uptake. Plants take in water from the soil, through their root hairs. Figure 8.9 shows the end of a root, magnified. At the very tip is a protective cap, to protect the root as it grows through the soil. The rest of the root is covered by a layer of cells called the epidermis. The root hairs are formed from some of the cells in the epidermis. You can see in Figure 8.9 that there are no root hairs at the very tip of the root, they start a little way behind it. Each root hair is a long epidermal cell. Root hairs do not live for very long. As the root grows, they get damaged by the soil particles, and are replaced by new ones. Figure 8.9, a micrograph of the tip of a root, magnified 70 times. Figure 8.10, a photomicrograph, showing part of a transverse section of a root. You can see several root hairs on the right-hand side. Figure 8.11, how water moves from the soil to the xylem vessels in a root. The function of root hairs is to absorb water and mineral ions from the soil. We have seen that water moves into a root hair by osmosis. The cytoplasm and cell sap inside the root hair are quite concentrated solutions. The water in the soil is normally a more dilute solution. Water therefore diffuses into the root hair, down its concentration gradient, through the partially permeable cell surface membrane. The long, thin root hairs have a large surface area, which increases the uptake of water and mineral ions. The pathway of water through a plant. The root hairs are on the edge of the root. The xylem vessels are in the center. Before the water can be taken to the rest of the plant, it must travel across the root to these xylem vessels. The path that the water takes is shown in Figure 8.11. The water travels by osmosis through the cortex, from cell to cell. Some of it may also just seep through the spaces between the cells, or through the cell walls, never actually entering a cell at all. Eventually it reaches the xylem vessels in the middle of the root. These transport it all the way up through the stem, and into the leaves. When water reaches the xylem, it moves up xylem vessels in the same way that a drink moves up a straw when you suck it. When you suck a straw, you reduce the pressure at the top of the straw. The liquid at the bottom of the straw is at a higher pressure, so the liquid flows up the straw into your mouth. The same thing happens with the water in xylem vessels. The pressure at the top of the vessels is lowered, while the pressure at the bottom stays high. Water therefore flows up the xylem vessels. In the next topic, we will see what causes the reduction in pressure at the top of the xylem vessels. Questions. 6. What are the functions of root hairs? 7. Explain how the structure of a root hair helps it to carry out its function. 8. Use your knowledge of osmosis to explain how water moves from the soil into a root hair. 9. Use your understanding of water potential to explain how water moves from the soil and across the root cortex. Transpiration. Transpiration is the loss of water vapor from a plant. Most of this loss takes place from the leaves. You may remember that there are openings on the surface of the leaf called stomata. There are usually more stomata on the underside of the leaf, in the lower epidermis. The mesophyll cells inside the leaf are each covered with a thin film of moisture. Some of this film of moisture evaporates from the cells into the air spaces. This water vapor diffuses out of the leaf through the stomata. Water from the xylem vessels in the leaf then travels to the mesophyll cells by osmosis to replace it. Water is constantly being taken from the top of the xylem vessels to supply the cells in the leaves. This reduces the effective pressure at the top of the xylem vessels, so that water flows up them. This process is known as the transpiration stream. Figure 8.12, the transpiration stream. Figure 8.13, how water moves from xylem to the air, through a plant leaf. Experimental skills 8.1. Investigating the pathway of water through the above-ground parts of a plant. In this investigation, you will use a colored liquid to trace the path of water, as it moves through a plant stalk and leaf. You will practice following instructions, observing carefully and drawing conclusions. You can also try planning another experiment using the technique you have just used. You will need a celery stalk, with some leaves attached. A small beaker. Some water with a colored dye added, such as eosin. A sharp knife or scalpel. A surface on which you can cut the stalk. A hand lens. Safety, take care with the sharp knife or scalpel. Try not to get the dye on your skin or clothes, as it will be very difficult to wash off. Method 1. Put a small quantity of the colored water into a beaker. 2. Stand the celery in the colored water. Leave it for at least an hour longer if you can. 3. Remove the celery from the water. Place it on a tile and cut it across. Study the cut end with a hand lens. It may look like the one in figure 8.14. 4. Make a labeled drawing of the surface of your celery stalk. 5. Look at the leaves. Can you see the dye in the leaves? If so, describe which part contains the dye. Questions. 1. From the position of the colored areas in the stalk in figure 8.14, and in your own celery stalk, which tissue in the stalk do you think contains the colored water? 2. Did the colored water get into the leaves? If so, which part of the leaf contains the colored water? 3. Suggest how you could use this technique to test this hypothesis. Water moves more quickly through a plant when the temperature is higher. Think about the variable you will change and how you will do this. The variables you will try to keep constant, and how you will do this. How and when you will measure your results. Any safety risks in your experiment, and how you will minimize them. Figure 8.14. A cut celery stalk that has been standing in a solution of a red dye. Activity 8.2. How can we use a balance to estimate rate of transpiration? Figure 8.15 shows two plants in pots. One plant has a transparent plastic bag covering the whole plant and pot. The other plant has a transparent plastic bag covering only the soil and pot. Figure 8.15. Using pot plants to measure the rate of transpiration. In your group, discuss how you could use these plants and a balance to measure how quickly a plant is losing water vapor by transpiration. Be ready to share your ideas with the rest of the class. Transpiration Pull. The spongy mesophyll tissue in a plant leaf is very important in helping to keep water moving through the plant. The combined surface area of all the spongy mesophyll cells is very large, and this surface is in contact with the air spaces in the leaf. Liquid water moves into the mesophyll cells from the xylem vessels by osmosis. A lot of this water then evaporates from the cell walls, ending up as water vapor in the interconnecting air spaces. As we have seen, the water vapor then diffuses out through the stomata and into the air surrounding the leaf. The rate at which this water vapor diffuses out of the leaf is generally greater in leaves where there are many open stomata. This constant movement of water from the xylem, through the mesophyll cells, into the air spaces, and through the stomata into the air means that water is removed from the upper ends of the xylem vessels. This reduces the pressure in the vessels. The pressure at the top of the xylem vessels is therefore less than the pressure at the bottom, in the roots. This pressure difference causes water to flow upwards, from higher pressure at the bottom to the lower pressure at the top. The pressure difference is called transpiration pull, because it is caused by the loss of water vapor by transpiration. You can think of transpiration pull as similar to sucking a drink through a straw. As you suck at the top of the straw, you reduce the pressure. The pressure at the bottom of the straw is greater than at the top, so the drink flows up the straw and into your mouth. The drink in the straw, and the water in the xylem, can flow upwards like this because water molecules have a strong tendency to stick to each other. There is a force of attraction between them. As one water molecule moves upwards, others stick with it and move up as well. The water therefore stays together, as one continuous column, and does not break apart. Questions. 10. Put these words in order to describe the sequence in which water moves through a plant. Mesophyll cells, root cortex cells, root hair cells, xylem. 11. State whether water is in the form of a liquid or a gas. In each of these places, As it moves from the soil into root hairs, Bas it moves through xylem vessels, Cas it moves out of plant leaves into the air. 12. Explain the importance of the large surface area of the mesophyll cells in helping water to move through a plant. 13. Explain, in your own words, why water moves up through xylem vessels in an unbroken column. Reflection. It can be tricky to explain why water moves through a plant, because the process that causes it, transpiration, happens at the end of the pathway, not at the beginning. Do you find it easier to describe and explain this movement by starting at the top with transpiration, or starting at the bottom with root hairs? Try to decide why you find one way easier than the other. Use your decision to plan how you would manage if a question asks you to describe it the other way round. Measuring transpiration rates. It is not easy to measure how much water is lost from the leaves of a plant. It is much easier to measure how fast the plant takes up water. The rate at which a plant takes up water depends on the rate of transpiration. The faster a plant transpires, the faster it takes up water. Figure 8.16 illustrates apparatus, which can be used to compare the rate of transpiration in different conditions. Figure 8.16, a potometer with a reservoir, which makes it easy to refill. It is called a potometer, which simply means water measurer. By recording how fast the air-water meniscus moves along the capillary tube, you can compare how fast the plant takes up water in different conditions. There are many different kinds of potometer, so yours may not look like figure 8. 16. The simplest kind is just a long glass tube, which you can fill with water. A piece of rubber tubing slid over one end allows you to fix the cut end of a chute into it, making an airtight connection. This works just as well as the one in figure 8.16, but is much harder to refill with water. Figure 8.17, simpler potometer without a reservoir. This one can be refilled by taking it apart and submerging the tube in water. Experimental skills 8.2. Using a potometer to investigate the effects of different variables on the rate of transpiration. In this investigation, you will use a potometer to measure how fast a plant shoot takes up water in different conditions. You will practice handling apparatus carefully, making accurate measurements, recording results and drawing conclusions. You will also evaluate your method and think about possible improvements. You will need a potometer, like the ones in Figure 8.16 or 8.17. A retort stand, boss and clamp to support the potometer. A fresh, leafy shoot. A sharp blade to cut the end of the shoot so that it fits into the potometer. A timer. A fan. Access to places with different temperatures, for example a warm room and a fridge. Safety, take care with the sharp blade. In Step 2, put the plant shoot onto a non-slip surface such as a corkboard to cut it. Cut away from you, not towards you. Before you start, look carefully at your potometer and check you know how it works and how to use it. Method 1. Fill the potometer with water. If you have a potometer like the one in Figure 8.16, you can simply open the clip and then close it once the tubes are all full of water. If it is like the one in Figure 8.17, submerge it in a big container of water. 2. Cut a plant shoot, making a slanting cut. Push the shoot into the potometer. If your potometer is like the one in Figure 8.17, do this while the tube is still completely under water. 3. If you have submerged your potometer in water, take it out of the water and stand it on the bench. Check that your potometer is airtight and that there are no air bubbles. 4. Now leave the apparatus in a light, airy place. Fix the ruler close to the tube, to act as a scale. As the plant transpires, the water it loses is replaced by water taken up through the cut end of the stem. Air will be drawn in at the end of the capillary tube. 5. When the air-water meniscus reaches the scale, begin to record the position of the meniscus every 2 minutes for 10 minutes. 6. Repeat the investigation, but with the apparatus in a different situation. You could try each of these. Blowing it with a fan. Putting it in a refrigerator. Questions 1. A. Did you have any difficulties setting up the potometer and getting the meniscus to move? If so, explain what you think caused the difficulties. B. Did you manage to overcome the difficulties? If so, describe how you did this. 2. Explain why the air-water meniscus moves along the scale. 3. Use your results to draw line graphs, so that you can easily compare the rate of movement in different conditions. Conditions that affect transpiration rate. The rate at which water vapor diffuses out of a plant's leaves is affected by the environment. Transpiration happens faster when the temperature is high and when it is windy. The higher the temperature, the greater the kinetic energy of water molecules. This means that water evaporates faster from the surface of the mesophyll cells, and the water vapor diffuses out of the leaf into the air more quickly. On a windy day, the air around the leaf, which contains a lot of water vapor that has just diffused out of the leaf, is quickly moved away. This means that there is always a diffusion gradient for the water vapor, because there is less of it outside the leaf than in the air spaces inside the leaf. So, water vapor diffuses out of the leaf faster on a windy day than on a day when the air is still. Transpiration rate is also affected by humidity. Humidity means the moisture content of the air. The higher the humidity, the less water vapor will diffuse out from the leaves. This is because there's not much of a diffusion gradient for the water between the air spaces inside the leaf and the wet air outside it. Transpiration decreases as humidity increases. If the temperature is high, it is windy and the air is very dry, then transpiration will happen very quickly. The plant may lose water from its leaves faster than it can take it up from its roots. The individual cells in the plant lose so much water that they become flaccid. The tissues in the leaves are no longer supported by the turgid cells pushing outwards against one another. The leaves become soft and floppy. This is called wilting. Figure 8.18. These pictures, taken over a period of 1 hour, show a plant that is gradually losing more water by transpiration than it can take up into its roots. Questions 14. Copy and complete this sentence. Transpiration happens faster when temperature is and wind speed is. 15. To measure transpiration rate using a potometer, we need to record two values. One is the distance moved by the meniscus. What is the other? 16. Explain why a plant is more likely to wilt on a hot day than on a cold day. 17. Look at the wilted plant in Figure 8.18. Suggest how wilting might help a plant to survive the shortage of water that has caused it to wilt. 8.3. Translocation of sucrose and amino acids. Leaves make carbohydrates by photosynthesis. They also use some of these carbohydrates to make amino acids, proteins, fats and oils and other organic substances. Some of the substances made in the leaves, especially sucrose and amino acids, are transported to other parts of the plant in the phloem tubes. This is called translocation. Sources and sinks. The part of a plant from which sucrose and amino acids are being translocated is called a source. The part of the plant to which they are being translocated is called a sink. When a plant is actively photosynthesizing and growing, the leaves are generally the major sources of translocated material. They are constantly producing sucrose, which is carried in the phloem to all other parts of the plant. These receiving parts, the sinks, include the roots and flowers. The roots may change some of the sucrose to starch and store it. The flowers use the sucrose to make fructose. Later, when the fruits are developing, sucrose may be used to produce sweet, juicy fruits ready to attract animals. But many plants have a time of year when they wait in a state of reduced activity for harsh conditions to end. In a hot climate, this may be during the hottest, driest season. In temperate countries, it may be during the winter. During these difficult times, the plant does not photosynthesize. It survives by using its stores of starch, oils and other materials in its roots. The stored materials are converted to sucrose and transported to other parts of the plant. So, these storage areas have now become sources. For example, baobab trees grow in tropical countries such as Madagascar. In the wet season, their leaves photosynthesize and make sucrose. This is transported to the trunk and roots, where it is stored as starch. In the dry season, the baobab drops its leaves. When it rains again, the stores of starch are changed to sucrose and transported to the growing buds, helping them to grow and form new leaves. Figure 8.19, baobab trees in the wet season, when the leaves are sources of sucrose. You can see from this example that phloem can transfer sucrose in either direction, up or down the plant. This isn't true for the transport of water in the xylem vessels. That can only go upwards, because transpiration always happens at the leaf surface, and it is this that provides the pull to draw water up the plant. Questions. 18 inches the wet season, which part of a baobab is a source, and which parts are sinks? 19 as the dry season comes to an end, which part of a baobab becomes a source, and which parts are sinks? 20 phloem tubes can transport sucrose both up and down a plant. Explain why xylem can only transport water up a plant and not down it. Figure 8.20, in the dry season, the stems and roots store starch, ready to supply the leaves with sucrose when they begin to grow in a few months' time. Summary. In plants, xylem transports water and mineral ions and supports the plant. Phloem transports sucrose and amino acids. In roots, xylem and phloem are found in the center, but in stems they are arranged in groups close to the outer edge. In leaves, they are found in the veins. Xylem vessels have thick, strong walls containing lignin, and no cell contents. Their cells are joined end to end with no cross walls. These features help them to carry out their functions of support and water transport. The large surface area of root hair cells helps them to absorb water and mineral ions. Water moves from root hair cells through the root cortex, through the xylem, and into the mesophyll cells in leaves. Transpiration is the movement of water vapor from the air spaces in a leaf into the air outside the leaf, through stomata. The water vapor in the air spaces comes from the mesophyll cells, as it evaporates from their cell walls. The loss of water vapor from the leaves reduces the pressure at the top of xylem vessels, and water moves up the xylem, as a result of this transpiration pull. Attractive forces between water molecules help the water to move in a continuous column. High temperatures and high wind speeds increase the rate of transpiration. High temperature increases kinetic energy of molecules, which speeds up evaporation and diffusion. High wind speed and low humidity increase the diffusion gradient for water vapor from the air spaces into the surrounding air. Plants wilt if they lose water faster than they can absorb it, so that cells lose their turgidity. Translocation is the transport of sucrose and amino acids from sources to sinks, in phloem tubes. Project: making a display about using eucalyptus trees to find gold. You are going to research information on the internet and use your findings to contribute to a display. Scientists have discovered that some eucalyptus trees have nanoparticles of gold in their leaves. The gold is present in the leaves that are growing on living trees, and also in the dead leaves that fall to the ground. Figure 8.21. Eucalyptus trees are helping mining companies to find gold that is deep underground. In your group, use the internet to find out more about this discovery. Choose one or more of these issues to research. Once you begin, you may also find another interesting issue that you would like to research, that is not in this list. Where in the world has this discovery been made, and how was the discovery made? Where does the gold come from? How do the trees absorb the gold? How is the gold transported to the leaves of the trees? Why are mining companies very interested in this discovery? How might the discovery eventually affect the environment where the trees grow? Try to share out the different areas of research between different groups, and plan how each group will contribute to the final display. Exam-style questions. 1. What is a function of phloem, but not xylem? A. Supporting the plant. B. Transporting amino acids. Transporting mineral ions. D. Transporting water. 2. Which word best completes the sentence? Water evaporates from the cells in the leaves and then diffuses out of the stomata. Epidermal bigard mesophyll. D. Xylem. 3. The diagrams show a transverse section through a root and a stem. Which label on the diagram of the stem indicates the same tissue as X? 4. The diagram shows an experiment set up by a learner. Which sequence shows the order of loss of mass by the leaves after 1 hour? Increasing loss of mass APQ, R, SBQ, R, P, SCR, Q, P, S. DSP, Q, R. 5. The diagram shows a plant that has had a ring of tissue removed from its stem. After 2 days, the concentration of sucrose at X was much greater than at Y. What explains this? A. Phloem has been removed, so sucrose cannot move from the sink to the source. B. The phloem has been removed, so sucrose cannot move from the source to the sink. C. The xylem has been removed, so water cannot move down the stem and transport the sucrose. D. The xylem has been removed, so water cannot move up the stem and dilute the sucrose. 6. A. Name the tissue that transports water from the roots to the leaves of a plant. A student set up the apparatus shown in the diagram. The student recorded the position of the air bubble every 2 minutes for 10 minutes. She then switched on a fan and recorded the position every 2 minutes for the next 10 minutes. The table shows her results. Time minutes. Position of air bubble millimeter. No fan. With fan. 0. 8. 10 to 11. 14 4. 15, 19:6, 18, 24:8, 21, 29:10, 25, 34. B. Explain why the air bubble moved up the capillary tube. C. With no fan, the mean rate of movement of the bubble was 1.7 millimeters per minute. Calculate the mean rate of movement of the bubble with the fan blowing. Show your working. D. State one factor, other than wind speed, that could increase the rate of movement of the bubble. 7. The figure below is a photomicrograph of the center of a root. A. Identify the tissues labeled X, Y, and Z. B. Describe how water is taken up into a root hair from the soil. C. Outline the pathway taken by water from the root hair to the leaves. 8. A. Leaves lose water vapor through their stomata. The micrograph shows a surface view of the lower epidermis of a leaf. The area of the leaf shown in the micrograph is 0.06 millimeters. Calculate the mean number of stomata per millimeter of the surface of the lower epidermis. Give your answer to the nearest whole number. B. Explain the importance of each of the following in the movement of water through a plant. I. Root hairs have a large surface area. 2. There are many interconnecting air spaces between the mesophyll cells in a leaf. 3. There are forces of attraction between water molecules. C. Describe 2 ways in which transport in phloem differs from transport in xylem. B. Water vapor diffuses out of a plant's leaves. Explain where this water vapor comes from. C. The cells in the leaves of a plant growing in hot conditions can become 9. Stomata open to allow carbon dioxide to diffuse into leaves. However, if the plant is short of water, stomata may close to prevent loss of water vapor. A. Explain why it is important to a plant to allow carbon dioxide to diffuse into its leaves. Partly dehydrated. This means that they contain less water than normal. The graph shows how the percentage hydration of a plant's leaves and the hydration of the soil change during a period of 5 days without any rainfall. I use the data in the graph to describe how the hydration of the leaves changes during the 5 days without rainfall. To suggest an explanation for the trend and pattern you have described in your answer to I. 3. Predict the appearance of the plant on day 5. Explain your prediction. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. I can see topic. Needs more work. Almost they're confident to move on. State the functions of xylem and phloem. 8.1 Identify the positions of xylem and phloem in roots, stems and leaves. 8.1 Identify root hair cells, state their functions, and explain how their structure is related to their functions. 8. 2. Describe the pathway taken by water from soil to leaves. 8.2 Investigate the pathway of water through the above-ground parts of a plant. 8.2 Explain what transpiration is and how it happens. 8.2 Investigate and describe the effects of variation of temperature and wind speed on transpiration rate. 8.2 Relate the structure of xylem vessels to their functions. 8.1 Explain the importance of the large surface area of the mesophyll cells. 8.2 Explain the mechanism that causes water to move up through xylem vessels. 8.2 Explain how temperature, wind speed and humidity affect transpiration rate. 8.2 Explain how and why wilting occurs. 8.2 Explain what is meant by translocation. 8.3 Explain why some parts of a plant may act as sources and sinks at different times. 8.3. Chapter 9. Transport in animals. In this chapter you will find out about the human circulatory system. Learn about the structure and function of the heart. Think about factors that increase the risk of developing heart disease. Investigate how exercise affects heart rate. Compare the structure and function of arteries, veins and capillaries. Find out about the components of blood and what they do. Explain how the structures of arteries, veins and capillaries are related to their functions. Getting started. Draw a very simple diagram of a human cell, showing its nucleus, cell membrane and cytoplasm. Draw arrows pointing into the cell, and label them to show what the cell needs to take in to stay alive. Draw arrows pointing out of the cell, and label them to show what the cell has to get rid of. Think about how these substances are brought to the cell and taken away from it. Share your ideas with the rest of the class. Blood transfusions. Every day, thousands of people worldwide need to be given a blood transfusion. This could be because they have had an injury and lost a lot of blood, or perhaps they have a disease that has caused a problem with their blood. The blood that is used for transfusions is given by blood donors. In most countries, donors give their blood for free, but in some countries, hospitals pay people for their donated blood. The donor is tested carefully beforehand to make sure that their blood does not contain anything harmful, such as viruses. Figure 9.1. A nurse takes blood from a blood donor. Collecting the blood takes about 10 minutes, but testing and checking the donor and their blood involves about another 20 minutes. The donor is also tested to find their blood group. There are 4 main blood groups A, B, AB and O. Blood group O can be safely given to anyone, but in general it is best to match the donated blood to the blood group of the recipient. Although donated blood can be stored at low temperature for a while, there is sometimes a shortage of particular blood types. For a long time, scientists have been trying to produce artificial blood, which could be safely given to anyone to keep them alive at least until there is enough real blood available for them. Progress is being made, and perhaps by the time you are reading this, artificial blood may be used in some countries. Discussion questions. 1. Do you think it is a good idea to pay donors for their blood, or is it better for blood to be given for free? 2. What might be the advantages of using artificial blood rather than blood taken from donors? 9.1. Circulatory systems. The main transport system of all mammals, including humans, is the blood system. It is also known as the circulatory system. It is a network of tubes, called blood vessels. A pump, the heart, keeps blood flowing through the vessels. Valves in the heart and blood vessels make sure the blood flows in the correct direction. Figure 9.2 shows the general layout of the human blood system. The arrows show the direction of blood flow. Put your finger on the position of the lungs, at the top of the diagram, and then follow the arrows. You can see that blood flows from the lungs into the left-hand side of the heart, and then out to the rest of the body. It then flows back to the right-hand side of the heart, before traveling to the lungs again. Figure 9.2, the general layout of the circulatory system of a human, as seen from the front. Oxygenating the blood. The blood in the left-hand side of the heart has come from the lungs. It contains oxygen, which was picked up in the capillaries surrounding the alveoli. It is called oxygenated blood. This oxygenated blood is then sent around the body. Some of the oxygen is taken up by the body cells, which need oxygen for respiration. When this happens, the blood becomes deoxygenated. The deoxygenated blood is brought back to the right-hand side of the heart. It then goes to the lungs, where it becomes oxygenated once more. Double and single circulatory systems. The circulatory system shown in Figure 9.2 is a double circulatory system. This means that the blood passes through the heart twice on one complete circuit of the body. We can think of the double circulatory system being made up of two parts: the blood vessels that take the blood to the lungs and back, called the pulmonary system, and the blood vessels that take the blood to the rest of the body and back, called the systemic system. Double circulatory systems are found in all mammals, and in birds and reptiles. However, fish have a circulatory system in which the blood passes through the heart only once on a complete circuit. This is called a single circulatory system, and is shown in Figure 9.3. Figure 9.3, the single circulatory system of a fish. Double circulatory systems have some advantages over single circulatory systems. When blood flows through the tiny blood vessels in a fish's gills or a mammal's lungs, it loses a lot of the pressure that was given to it by the pumping of the heart. In a mammal, this low-pressure blood is delivered back to the heart, which raises its pressure again before sending it off to the rest of the body. In a fish, the low-pressure blood just continues around the fish's body. This means that blood travels much more slowly to a fish's body organs than it does in a mammal. This is particularly important when you think about the delivery of oxygen for respiration. Any tissues that are metabolically very active need a lot of oxygen delivered to them as quickly as possible, and this delivery is much more effective in a mammal than in a fish. Questions. 1. Look at Figure 9.2. Which side of the heart contains oxygenated blood, and which contains deoxygenated blood? 2. Explain how, and where, blood becomes deoxygenated. 3. Explain why a double circulatory system can provide respiring cells with oxygen more quickly than a single circulatory system. 9.2. The heart. The function of the heart is to pump blood around the body. It is made of a special type of muscle that contracts and relaxes regularly throughout life. Figure 9.4 is a section through a heart. The heart is divided into 4 spaces, called chambers. The 2 upper chambers are called atria. The 2 lower chambers are ventricles. The chambers on the left-hand side are completely separated from the ones on the right-hand side by a septum. If you look at figures 9.2 and 9.4, you will see that blood flows into the heart at the top, into the atria. Both of the atria receive blood. The left atrium receives blood from the pulmonary veins, which come from the lungs. The right atrium receives blood from the rest of the body, arriving through the big veins called the venae cavae. Figure 9.4, a vertical section through a human heart, seen from the front. From the atria, the blood flows into the ventricles. The muscles in the walls of the ventricles contract. This pumps the blood out of the heart. The muscle contracts strongly, producing a large force that squeezes inwards on the blood inside the heart and pushes it out. The blood in the left ventricle is pumped into the aorta. This is a big artery that takes the blood around the body. The right ventricle pumps blood into the pulmonary artery, which takes it to the lungs. Valves. Look again at the one-way valves in Figure 9.4. The valves between the atria and the ventricles are called Atrioventricular Valves. They allow blood to flow from the atria into the ventricles, but prevent it from going in the opposite direction. The valves in the entrances to the aorta and the pulmonary artery are called Semilunar Valves. This name means half moon and refers to their shape. These valves allow blood to flow from the ventricles into the arteries, but stop it going the other way. Differences in muscle wall thickness. The function of the ventricles is different from the function of the atria. The atria simply receive blood from either the lungs or the body and supply it to the ventricles. The ventricles pump blood out of the heart and all around the body. To help them do this, the ventricles have much thicker, more muscular walls than the atria. There is also a difference in the thickness of the walls of the right and left ventricles. The right ventricle pumps blood to the lungs, which are very close to the heart. The left ventricle, however, pumps blood to the rest of the body. The left ventricle has an especially thick wall of muscle to enable it to do this. The blood flowing to the lungs in the pulmonary artery has a much lower pressure than the blood in the aorta. Questions. 4. Look at Figure 9.2 and Figure 9.4. List, in order, the parts of the heart that blood flows through on one complete journey around the circulatory system. 5. Which parts of the heart contain oxygenated blood? 6. Do arteries carry blood